Welcome to Click for IB Biology. Our topic of study is evolution and speciation, and this particular video is going to be looking at the evidence for evolution as laid out by DNA and RNA. There's a blank version of this presentation. That's a PDF, and you'll find that linked in down below in the description. And of course, as always, there's 10 questions there which you can then answer and check out your answers with those that I have provided. There's also a link to an AI prompt that will give you more and more questions, which is by far the best way to secure your learning. So let's take a look at the evidence for evolution as exists in the molecules DNA and RNA. And so with evolution over time and the change in the heritable characteristics of populations in that time, it's notable that for all known species, that DNA is the common genetic material. We would say that we would speak of the universality of DNA as the heritable material itself. And in and of itself, this would provide evidence of the success of DNA as the heritable material and of evidence of that evolutionary process, that DNA is so effective in encoding the genetic information for an organism that it has been retained and that there are no other ways of doing that. And of course, so we can trace our DNA all the way back to the last universal common ancestor at the where all contemporary and past species can find their origins. Not only is there a universality of the type of genetic encoding that exists, but also in the actual code system within that molecule. And so we have the universality of the genetic code, which is being conserved as well. And we see this in the conservation of codons, so that the same codons that we have, or triplets, code for the same amino acids. And we will have seen this as we have gone through our study on the structure of DNA. But beyond that, we can see that in the universality of the genetic code, how there's been a conservation of critical genes. And a famous one would perhaps be the cytochrome genes, in which one can compare the base sequence of those genes, and whilst it retains largely an unchanged codes, those minor modifications in different species reflect the divergence in the process of the speciation and the creation of diversity of life. But those critical genetic genes, those critical genes have been conserved by and large. Of course, any DNA RNA evidence would need to contain within it a mechanism to allow for changes in the heritable characteristics using the IB definition. And of course, the molecule DNA and indeed RNA accounts for the variation which is seen in populations. And in our study of molecular genetics, we'll have seen how things like single nucleotide polymorphism or point mutations account for mutation creating new alleles in a molecular mechanism. We've also seen how chromosomes can end up transverting and inverting and creating variation in that way as well. But of course, we also have the processes of meiosis and sexual reproduction, in which we have both the first and second Mendelian laws creating the tremendous variation, independent assortment, and so forth. And then beyond that, we then look into crossover, and then perhaps moving finally to random fertilization, and how these processes combine create the raw material for Darwin's mechanism of evolution known as natural selection. And so our evidence is not just in the universality of the molecule and its code, but in it allowing us to have a molecule which can change its the information as required by our theory of evolution, change over time. The proteome itself also exhibits evidence of evolution, in which there are molecular homologies, in which molecules, proteins, clearly inherited by a diversity of organisms, but in of themselves have been conserved. The protein structures are conserved in their amino acid sequence, and where there are differences, these small differences reflect the speciation process and the divergence of life through modification, as Darwin referred to descent through modification. And so the protein sequences, when compared, also provide us, like the genetic code, the proteome also provides us, in fact, with what one might call a molecular clock, that we can track the change of genetic information over time, and as such allows us to build trees of genetic diversity and evolution. In the 21st century, we have become very aware that even though DNA dominated the early part of the 20th century in its central position in what was then known as the central dogma, awareness of RNA's role in catalysis and in gene regulation and in protein synthesis, its role in protein synthesis, has now largely shifted attention from DNA back to RNA and its principal role in cell chemistry and the orchestration of molecular biology, the chemistry, the biochemistry of life. And it's RNA that will also, in the way its relatedness to DNA, allows us to find that evidence for evolution in its molecular functioning. And so these are examples of the roles that RNA and DNA play in the evolutionary evidence of change, the changes in the heritability of populations over time. And each of these topics is developed considerably in other topics on this course. And so this, in fact, is a summary of those points. In our next video, as you can see in the link on the left, we're going to be going on to look at the concept of homology and convergence. Please do subscribe to the channel, it's greatly appreciated. And on the right hand side is a quick list of all our evolutionary and speciation biology videos. Good luck in your studies.